Good morning. Today, let us discuss about cholesterol biosynthesis. What is cholesterol? Cholesterol is a waxy substance found in our blood. Our body needs cholesterol to build healthy cells, but levels of cholesterol can increase our risk of heart disease. Total cholesterol level under 200 milligram is healthy for adults. With high cholesterol, you may develop fatty deposits in our blood vessels. Cholesterol is found exclusively in animals, hence, it is often called as animal sterols. The approximate total body content of cholesterol in adult man weighing 70 kg is about 140 gram, that is approximately 2 gram per kilogram body weight. Cholesterol is amphipathic in nature since it possesses both hydrophilic and hydrophobic regions in its structure. Biochemistry of cholesterol molecule or its structure. Its molecular formula is C27H46O. It has 4 fused ring called styrene nucleus. It contains single double bond at position 5,6 carbon atom and contains a single hydroxyl group at third carbon. It also contains 2 methyl group at carbon atom 18 and 19. It has an acyl side chain at carbon atom 7. Let us go to the biochemical importance. Cholesterol is a structural component of cell membrane and lipoprotein like HDL and LDL. They are carriers of cholesterol in our bloodstream. Cholesterol is the precursor for the synthesis of all other steroids in our body. These include steroid hormones, vitamins A, D, E, and K. It also a precursor for the biosynthesis of bile acids. Fatty acids are transported to liver as cholesterol esters for oxidation and for the production of energy. The Nobel Prize in Physiology or Medicine was awarded jointly to Nordbosch and Feder-Lenin in 1964 for their discoveries concerning the mechanism and regulation of the cholesterol biosynthesis and fatty acid metabolism. What is the biochemical importance of cholesterol biosynthesis? Cholesterol is essential to life as it performs a number of important functions. About 1 gram of cholesterol is synthesized per day in adults. Almost all the tissues of the body participate in cholesterol biosynthesis. The largest contribution is made by liver, it is 50%, followed by intestine, skin, adrenal cortex, reproductive tissues, etc. The enzymes involved in the cholesterol biosynthesis are cytosolic and biosynthesis are found in cytosol and microsomal fractions of the cell. Acetate of acetyl-CoA provides all the carbon atom in cholesterol molecule. The reducing equivalents are supplied by NADPH while ATP provide energy for the cholesterol biosynthesis. For the production of 1 mole of cholesterol, 18 moles of acetyl-CoA, 36 moles of ATP and 16 moles of NADPH are required. Biosynthetic pathway. The synthesis of cholesterol can be learned in 5 stages. At first, the synthesis of HMG-CoA, it is a 6 carbon molecule. Then formation of mevalonate, it is also a 6 carbon molecule. Production of isoprenoid units, it is a 5 carbon unit. Then synthesis of squalene, it is a 30 carbon molecule. And the last stage, conversion of squalene to cholesterol. Let us discuss the stages in detail. Stage 1, the synthesis of HMG-CoA. Acetyl-CoA molecule acts as the precursor for the biosynthesis of cholesterol. The acetyl-CoA molecules are synthesized in the mitochondria from pyruvate by the enzyme pyruvate dehydrogenase enzyme complex. These acetyl-CoA passes through the cell membrane in the form of citrate. In the cytosol, this citrate is hydrolyzed or divided into two molecules, oxaloacetate and acetyl-CoA, and utilized for the biosynthesis of acetoacetyl-CoA. In the first stage, two molecules of acetyl-CoA condense together in the presence of thiolase enzyme after the removal of coenzyme A to produce acetoacetyl-CoA. Acetyl-CoA is a 2-carbon molecule. Acetoacetate is a 4-carbon molecule. That here it is the condensation of two molecules of acetyl-CoA. In the next stage, one molecule of acetyl-CoA again added to the acetoacetyl-CoA to produce a 6-carbon compound HMG-CoA or 3-hydroxy 3-methyl glutarate-CoA. It is a glutaric acid derivative, hence the name HMG-CoA. We can see the carbon atoms, carbon 1, 2, 3, 4, 5 and 6th carbon as a side chain methyl group. The major enzymes are thiolase and HMG-CoA synthase. The thiolase and HMG-CoA enzymes are cytosomal. Thiolase is an oxidant enzyme that removes electrons. The isoenzyme of HMG-CoA synthase is also present in mitochondrial fraction. The HMG-CoA synthase catalyze the biosynthesis of HMG-CoA, and these HMG-CoA molecules are utilized for the biosynthesis of ketone bodies. That means there are two isoenzymes of HMG-CoA. One is cytosolic and another one is mitochondrial. Then stage 2. Stage 2 is the synthesis of mevalonate from HMG-CoA. Both these compounds are 6-carbon compound. Here one of the important reduction takes place during this mechanism in the presence of NADPH. This NADPH molecule is provided by the pentose phosphate pathway. HMG-CoA reductase is the rate limiting enzyme in the cholesterol biosynthesis. That means it is a major regulatory enzyme in the entire cholesterol biosynthetic pathway. This enzyme is present in endoplasmic reticulum and catalyzes the reduction of HMG-CoA to mevalonate. HMG-CoA reductate transcription is tightly regulated by membrane-bound transcription factor designated as sterol regulatory element binding protein SREBP2. Then stage 3. Stage 3 is the production of 5-carbon unit isopentenyl pyrophosphate and dimethyl allyl pyrophosphate from mevalonate. In a 3-step reaction catalyzed by kinases, mevalonate is converted into IPP, isopentenyl pyrophosphate. It again isomerizes to DPP, dimethyl allyl pyrophosphate. Both IPP and DPP are 5-carbon isoprenoid units. You can see this, see the structure. Mevalonate, 5-carbon main chain and side chain methyl group. Therefore, it is a 6-carbon compound. After the 3-step phosphorylation, in the presence of mevalonate, 5-phosphotransferase and phosphomevalonate kinase, and again in the presence of enzyme pyrophosphomevalonate decarboxylase, it is converted into 3-phosphor 5-pyrophosphomevalonate. It is an unstable intermediate. It immediately converted into isopentenyl pyrophosphate after the decarboxylation. 3-phospho 5 pyrophosphomalonate is an unstable intermediate. Hence, it is represented in a square bracket. After the decarboxylation, it is converted into delta 3 isopentenyl pyrophosphate. Delta 3 representing the position of double bond at third carbon. It is isomerized to 5 carbon unit dimethyl allyl pyrophosphate. Here it is a change in double bonds. Okay. Both these intermediates are very important. 5 carbon isoprenoid units, IPP and DPP. In the next stage, stage 5, both the molecules of IPP and DPP condenses to produce 10 carbon geranyl pyrophosphate. 5 carbon molecule, 5 carbon pyrophosphate plus delta 3 isopentenyl pyrophosphate. 5 plus 5, 10. 10 carbon intermediate geranyl pyrophosphate is produced. It is an activated molecule. Then geranyl pyrophosphate condensed with another molecule of IPP in a head to tail fashion. Head to tail fashion means head of the molecule linked to the tail portion of the another molecule. Tail portion representing the phosphorylated methyl group. Phosphorylated end or representing the tail portion. Geranyl pyrophosphate and isopentenyl pyrophosphate linked together to form pharencyl pyrophosphate. Pharencyl pyrophosphate is a 15 carbon molecule containing 15 carbon atom in its structure. In the next stage, another molecule of pharencyl pyrophosphate condensed together to form squalene. Squalene is a 30 carbon intermediate. That means 50 carbon pharencyl pyrophosphate, one molecule linked with another molecule of pharencyl pyrophosphate. It is also a 15 carbon. Then 15 plus 15, 30 carbon intermediate is formed. It is squalene. Squalene is one of the important intermediate in this pathway. In the last stage, squalene molecule begins the cyclization, oxidation, cyclization and loss of 3 methyl group result in conversion of squalene to cholesterol. In the first stage, squalene monooxygenase catalyzes the conversion of squalene to squalene 2,3 epoxide in presence of reducing equivalent NADPH. NADPH is provided by the pentose phosphate pathway. In the next step, cyclase enzyme in animals catalyzes the cyclization of the ring to produce lanosterol. Then lanosterol is converted to cholesterol through a number of series of reaction to produce a 27 carbon molecule cholesterol. Squalene 2,3 epoxide. Squalene 2,3 epoxide is also the precursor for the stigmasterol and ergosterol. Plant sterols, stigmasterols and fungal sterol, ergosterol. It is one of the important intermediate in the steroid biosynthetic pathways. Let us conclude the biosynthetic pathway. Again, starting with acetyl-CoA molecule 2 carbon, then 2 HMG-CoA 6 carbon, which is again converted into mevalonate. It is also a 6 carbon compound. In the next stage, after the decarboxylation, Mevalonate is converted into isoprenoid unit. Isoprenoid unit means it contains 5 carbon unit. It is a building block for a number of biomolecule. In the next stage, 6 molecules of isoprenoid unit condense together to produce squalene. It is a 30 carbon compound. In the next step, squalene is converted into lanosterol after oxidation, cyclization, and removal of the methyl group to lanosterol and to cholesterol. Other natural products derived from activated isoprene unit. Isopentenyl pyrophosphate is one of the major intermediate, major precursor molecule for the biosynthesis of a number of natural compounds like vitamins, especially fat-soluble vitamins A, D, E, and K. Then carotenoids, plant hormones, abscisic acid and gibberellic acids. Then a number of isoprenoid compound. Then quinones. Quinones are electron carriers, especially in electron transport chain and photosynthetic pathway. Ubiquinone and plastoquinone. In plants, it is termed as plastoquinone, and also for the biosynthesis of dolichols, and also for the phytol chain of chlorophyll, and it is also for the used for the biosynthesis of rubber, natural rubber. Then cholesterol act as the precursors for other steroid hormones and bile acids. Thank you.